a problem that physicists like to deal with. So I'm just going to show you a little bit about what we do and kind of give you a brief overview, but leave a lot of time for discussion. So my lab focuses on intrinsic activity. So if you think of, everything the brain does, the intrinsic activity is like the ocean. And these little blips that we look at that are related to task or stimulation, they're kind of like one wave on top of that ocean, right? So we want to characterize the whole body of water, the currents that are moving it, all of these things that are happening kind of underneath the surface of those little blips, because the intrinsic activity of the brain influences what you get in response to a task or stimulation. So this whole system works together. And when we do, kind of standard fMRI analysis and we average across different presentations of stimuli, we're basically getting rid of all that intrinsic activity. So some of the variability that we see is actually accounted for by what's going on throughout the rest of the brain when these particular tasks or stimuli are given. So we think that intrinsic activity is important, it's big, and it persists, across lots of different conditions, including in anesthetized animals. So whatever it is, there's something very fundamental about this and about its organization that we think is important to understand so that we can better interpret the changes that we see during task or stimulation. So I said the brain is a complex system. And what I mean by that is that activity occurs at multiple spatial and temporal scales. I mean, you can think of something going on at a single synapse, a single action potential. Then we've got kind of the mesoscale organization in columns and into layers that have specific inputs and outputs. And we have functional areas. And then, we also have small nuclei that can modulate large areas of the brain, driving changes in attention and arousal. And all of this stuff is kind of nested together. And when we look at resting state fMRI, it all gets mashed down into, one time course per voxel. So part of my lab's focus is to try to understand what we can pull out of this signal and relate back to particular features of the brain. I'm not really going to talk about that part today. I'm really just going to talk about, how we can go about characterizing this signal. How do we make sense of this intrinsic activity? What do we use to look at it, and how do we interpret what we see? So we got interested in this a long time ago. Actually, at the time, we were very skeptical about the whole resting state fMRI thing and set out to prove that it was entirely noise. And then we're, kind of surprised when we found out it wasn't entirely noise. It's good to be wrong sometimes. But one of the things we did once we were convinced that there was something in there is we believe that there ought to be changes, right? That, this activity is not steady. We ought to see some sort of dynamics. And so I was talking with my first PhD student and I said, well, go look at, what happens over time. And in my head, I had something like sliding window correlation in mind, something to give us some measurement of how areas were interacting with each other. But what he did was make a movie. And when he made a movie, this was data from anesthetized rats that we were looking at the time. When he made this movie, we could see these waves of activity that happened repeatedly over the course of the scan. And we said, wow, that's really weird. We ought to characterize that. And so he made a pattern finding algorithm to go in and kind of pull out these similar instances of occurrence. And, we weren't too excited about it, that it was probably related to anesthesia, until he put some of his human data, just for fun, through the same pattern finding algorithm. And out popped the same sort of reproducible, patterns of activity that occurred over and over throughout the scan. So we called those quasi-periodic patterns, just meaning that we see them a lot, but not necessarily all the time. And so I'm going to show you, this is the first algorithm that we use to characterize these. So we have this functional scan over time. You can assume each one of these images here is a volume, and so volumes over time. And in our first iteration, we originally picked out a pattern that we could see in the rats. It turned out later that you don't have to pick a pattern you can see. You can start with random seeds and everything works just fine. So you take a random seed, you pull out a template, and we have to assign a link to that template. It turns out in humans that the template's about 20 seconds long. We've tried different, links, and I can tell you about that another time. But we pull out that chunk, that chunk that we call the template, that 20-second chunk, and we say, okay, let's correlate that with the whole scan. What else looks like this chunk in the scan? And so when we look at the correlation time course over here, we see some peaks. And we go, oh, okay, well, those are times that also look like that same pattern. So we grab those chunks too, we average them together, and we make a new template. And then we just keep repeating that until the template converges and doesn't really change much. So the first iteration of this, like I said, started with random seeds. Instead of just one random seed, we usually did something like, oh, I don't know, 50 per scan, and then did some clustering just to make sure we didn't get some unrepresentative pattern for some reason. Our more kind of refined iteration of this actually used every single time point and then clustered to get the most representative pattern. Didn't really change our results much, but it was kind of appealing in that we were sure we weren't missing anything. But that's our quasi-periodic pattern finding algorithm. And so what we get out of this is this pattern template, which is volume over time, so 4 dimension, 20 seconds worth of time. And then the sliding window correlation vector that's approximately the length of the scan and tells us when that pattern is strong or weak. So if you wonder what the template looks like, this is what we get from the Human Connectome Project data. So here we're showing just the signal intensity in the template with every area scaled to have a mean of 0. So warm is above 0, cool is below 0. And you can see that when we start, the brain is kind of divided into these two networks. There's, the blue network and the orange network. The blue is kind of default mode like, and we call everything else task positive. So when I play this, you see over time that there's an alternation. The areas that were originally high become low, the ones that were low become high. And there's also propagation along the cortex in some areas. And look specifically kind of here along the medial areas, you can see this kind of propagation. And then sometimes I think up here in the cortex is another place that you can kind of see it moving along the surface of the cortex. So that's what these templates look like. And you can see over on the right, they're also present in subcortical regions. They're not as strong. So we made another movie where we zoom in just on the subcortical region, so you can see it a little bit better. Oops, it didn't go. Let me try again. There, and what you see is there's this same sort of two-system organization, and again, some propagation along particular structures, especially. So whatever this pattern is, it involves the whole brain. Subcortical, cortical, cerebellum, brainstem, all of this stuff is organized into this sort of cyclical anti-correlation of these two main features. Now, what we can do, besides just doing this algorithm once, is we can do the algorithm, pull out that quasi-periodic pattern, and then run the algorithm again. And if we do that, we can obtain multiple QPPs. So this, here we have the first one, second, third, fourth, fifth, sixth. Can you guys see my mouse? I've been assuming you can. Okay, good, good. And you can see when you're getting down here to 4, 5, and 6, these are getting pretty noisy, right? And I don't know if it really makes sense to actually calculate a QPP at that point. But that said, we can still pull out these patterns. So a lot of the structure of what we see in the intrinsic activity of the brain is accounted for by these patterns. And so to try to understand how much these patterns contribute to that activity that we see, we kind of work backwards. So we took the template that we got from our QPP algorithm. We took that correlation time course that we had that told us when it was strong. We did the convolution of those two. And then we tried to regress that out of the original signal. And when we did—
Excuse me.
Yeah.
I have a question.
Go ahead.
So can you go back to the previous slide? Then I think in your the GIF, you have like 30. I guess that's 30 time points, right?
Yes.
So does that imply that this 30 is like the length of a complete cycle?
Yes, that's
Oh,
about right for humans. It's about 20 seconds.
oh, so this is like biologically determined rather than determined by your model or algorithm, right?
Yeah, it is absolutely. And so we tried doing, different window lengths. And in general, if we use this algorithm and we do shorter windows, we get a partial pattern. If we do longer windows, we get multiple cycles. So it doesn't
Oh,
change
okay.
the underlying frequency of what we see. And I've got another method coming up that I'm going to show you because we always did have this question about window length. So we'll
Oh,
come back
okay.
to that. But yeah,
Oh,
exactly.
but
And I
does
should point
that?
out, oh,
Oh,
sorry,
okay.
one more thing, and then I'll come back to you.
Oh,
I should point out that the lengths of these cycles are different for different species. So
yeah,
for
I was
mice
about
and
to ask
rats,
that.
okay, perfect. I read your mind. So for mice and rats, they're on the order of 5 to 10 seconds, depending on the anesthetic that we use. And there's definitely an anesthetic influence, and we don't yet know why, whether it's neural activity or if it's something about the vasculature system. Okay, did I answer your question now?
Yes, thank you so much.
Okay. Okay, so we were trying to look at how much these patterns contribute to just, the intrinsic brain activity. And so we looked at resting state functional connectivity, so just correlation between different parcels in the brain before and after we did that regression. And so this is a correlation matrix. These are sorted into, 7 main networks. And this top right triangle is the raw data without us doing anything. Bottom left is the same data after we've taken out the primary QPP. And when we take out this primary QPP, you see that a lot of this anti-correlation becomes weaker. We lose a lot of the within network correlation. So we're accounting for quite a bit of that functional connectivity just with that first QPP. And then if we take out 2, we lose more. And by the time we get to 3, there's very little functional connectivity left. It's like these 3 spatiotemporal patterns actually account for most of the structure that we see. Now, there's a really intriguing exception to that, and that is this line, which represents the connections between left and right areas that are the same, like left and right motor cortex, left and right somatosensory cortex. So the mirrored analogs in the 2 hemispheres. They show much stronger connectivity even after QPP regression than anything else. So very intriguing that. Okay, so, that was the QPP algorithm. And we thought that was great. It told us a lot about, what's actually in this data that we're looking at. But we always had questions about the window length because we had to set that ahead of time. And one of the things that makes it mathematically a little bit iffy that you guys will probably appreciate better than I did is that, these aren't necessarily orthogonal. We're pulling out different components by regression, but we're not really enforcing orthogonality in any way. And so sometimes that's harder to interpret. So one of my postdocs wanted to look at this from a different perspective. So he did the same sort of analysis of intrinsic activity, but using some different tools. And what he noticed is that if you looked at those QPPs over here, there's kind of areas that just alternate in activity and areas where we see that propagation, right? So that made him think of these waves, and you can classify waves in a couple of different ways, but one is standing waves versus traveling waves. And so if you have a traveling wave, 2 points, 2 spatial points for us, have a constant relationship in time, but one might be stronger than the other at any given time. They have a time lag between their activity. Whereas, sorry, I gotta see if I can make this play again. Whereas if you have a standing wave, they always go up and down together in this sort of time lock fashion. So we felt like we saw aspects of both of those types of waves in our data, the kind of this alternation of activity in areas and also this propagation along the cortex. And so these are phenomena that are studied extensively by the climate community. And so he borrowed a tool that they use that's called, let's see if I can get this to move on. There we go. Oops, it is a complex PCA is what we're calling it. But basically you take the Hilbert transform of the data and then you do PCA. And what you get out of that then is the sort of spatiotemporal pattern with a phase that tells you when the pattern occurs at any time. And I've got a toy example here that I think makes it clearer. So here he's got a propagating wave. So this is at time point 5.8 seconds. We've got high activity up here. You know, this is just a simulation. So at like 8 seconds, it's moved down by 10 seconds at the bottom of the screen. And so he did complex PCA on this. And the first principal component pulls out a wave with this amplitude, so the right width, and this pattern of high versus low signal, and a phase lag. And the phase lag tells you, you know, when that wave hits different parts. So you can see that the phase here is much lower than the phase down here, showing that it's delayed. So that helped us to kind of get a grip on, you know, what this technique should pull out. And so we applied that. Go ahead. What was that?
Sorry. So where do you get the complex component from? I mean, is this from the fMRI data or?
This was just a toy example.
Sorry.
So he just added some noise, but now, yes, from the fMRI data. So we use that same Human Connectome Project data set, and he did 50 subjects, and again, did the Hilbert transform to turn it complex, then did PCA, and we looked at the different components.
Oh, I see.
And,
so you're, sorry. So the the complex data is coming from the Hilbert transform, but you still have magnitude fMRI data. Is that right? Okay.
Yes, we're not using the phase MRI data. Just the magnitude doing the Hilbert transform. Yes.
So I was just wondering, I mean, this is just discussion. I wonder if you have access to the phase data also, and you put both of them in the Hilbert transform, what it will look like. But of course, you haven't tried it. My question, this is just a suggestion.
No, I would like to know. I think that's a great suggestion, and I would like to know. I think that's very intriguing. We're going to have to get some of our own data so we can play with it and not have to deal with, you know, all of the stuff that they've already done to the HCP.
Sure. Yeah, thanks.
So I'm just going to kind of skip over these because I have movies that I think show them better. I did want to show this chart though. So if you look at the complex components, the first three kind of account for most of the variance, and then you start to see the sort of elbow drop off. So we're only looking at the first three. And the other thing I want to note is that we did not do global signal regression in this data. And if you work with fMRI or you know, at all, you know that that's a controversial thing. Whether if you do it, people will tell you not to. If you don't do it, people will tell you you should. So we left it in for this, and you're going to see that come out as its own particular component. Okay, so when we play these patterns based on the phases that we get from the complex principal components, they end up looking very much like the QPP. This first one is actually more global, and there have been some studies that looked at the global signal, and it looks very similar. Here's the second one now. This is back to our original QPP, which I should point out is normally obtained from GSR data. So global signal regressed. And then there's pattern 3, which I'm not really going to talk about today, except to say that it matches functional connectivity gradients. So if there is a method where people look at the functional connectivity of each independent voxel and how similar it is to other voxels, and they pull out, you know, kind of these low rank approximations that describe that and call them functional connectivity gradients. And for those, they look just like the QPPs, and you can pull out, you know, 5 or 6 of them. And our pattern 3 looks like the third one or so of the functional connectivity gradient. So we're picking up structure other people have seen before, but we don't really know how to interpret that one as well. So this first one, he's labeled this kind of strangely because we sometimes do QPP with global signal. If you do that, you get the same pattern. So the first complex component looks exactly like the spatiotemporal evolution of the global signal. And that one accounted for most of the variance. So we think that's what that is. So our second one then, like I said, is really, we think, what we were calling the QPP, the primary QPP. So now we have this notational problem too, on top of everything else, right? Now, because component 1 is global signal, component 2 is QPP1. It's very, very complicated, and we need to go in and clean that up at some point. But these two look very similar. So this is pattern 2, and I think the QPP was right before that. It might come up again. Let's see. Maybe I need to replay it. Here. Okay, yeah. So this one is obtained with the standard QPP algorithm. And now we should be switching over to pattern 2 from the complex PCA. So they have a lot of similarities. And there are some subtle differences, and we haven't explored those completely. And one of those is that propagation. There's more propagation in our traditional QPP algorithm than we see in some of these components, although they do capture propagation as well. So that's just kind of an open question. So that's where we are. That's what we've got. What I want to mention is that we have seen the same organization other ways. And one of the ways that I didn't put in today that I should have, because I sent the paper to Julia, is that we've taken fMRI data and we've fed it through an autoencoder. Now, basically, I'm not a serious deep learning person, but what an autoencoder does is it takes the data down and kind of projects it to a smaller space and then reconstructs it from that space. You can kind of think of it as like, taking your principal components and then throwing out all of the ones, that account for less than a certain amount of variance, right? It kind of cleans up your signal. So we did that, but what we were really interested in were the components that the autoencoder learned. And we forced it to learn spatiotemporal components that were about 20 seconds in length. And sure enough, out popped the same patterns, the same QPPs. So a completely different approach. We find the same structure in the data. The other thing I should say is that even though the complex PCA does not impose a window length, the same primary frequency dominates those waves. So if you look at like a power spectrum, they peak at around 0.05 hertz. And, so the pattern is approximately 20 seconds long. Sometimes it's a little stretched, sometimes it's a little compressed, but it's very close to that 20-second mark. So, the last thing I just wanted to put up here is we've kind of got too many ways to look at fMRI data, right? And there's a new analysis method coming out all of the time. And so I think we really need to try to unify how we think about the data, because every one of these analysis methods lends itself to a particular interpretation, right? So if you look at functional networks and then in a patient group, you see that one area is not connected, you say, oh, well, there's a disconnection in this network. But if you consider that functional network part of a larger pattern, that may not be where the disconnection is. It may not be between the areas where, that appear to be uncorrelated now. It may come from, some other type of input that's actually driving those areas. So I think we need to be careful about trying to find the right model and, thinking about how these different analysis methods interact. So I'm not going to go through this in any detail because it's just seriously painful, but it's in Taylor's paper. And what we did was we did 12 different analysis methods, from that smorgasbord of ways that we could look at the data. And the part I'm going to highlight is that we found that the main results from each method lined up to one of our 3 spatiotemporal patterns, or 3 complex PCs, which says to us that those 3 patterns really account for, a lot of the structure that we see in other things. So, we feel good about thinking of those as kind of the underlying model so far. We haven't found a better one. And, we think that we need to consider other analysis methods, say coactivation patterns, in the context of these large recurring spatiotemporal patterns that seem to be driving things. So that is, the end of my talk. So just to summarize, the 3 patterns that we found seem to account for most of the structure that we see with any analysis method for this intrinsic activity. And so that should make us think, where are these patterns coming from? How do you get this kind of repeated organization in the brain? It's not cognitive, right? I'm not having the same thought every 20 seconds. You know, there's other stuff going on. And if we want to know how the brain works, I think we need to figure out, you know, what that is. But even with these 3 patterns, you know, they account for most of the structure. I think it's like 70% of the functional connectivity, but only 1/3 of the variance. So there's still a lot of variance in the fMRI signal that's not accounted for. And I think it's fair to wonder if some of the activity that we're more interested in, you know, kind of brief transient activations in response to a stimulus, you know, in response to cognition, this sort of aperiodic stuff, if that might not show up in this other part after we take out these large scale patterns. But on the other hand, we've always got noise. And so I think a substantial amount of that variance has got to be noise for fMRI. There's just no getting around it. But I think there's room for noise and for really informative bits, you know, because we know it's there from fMRI, right? We know you can see changes related to cognition. So I think that must be in there too. I think we just have, you know, a really sticky problem of how to disentangle all of these different components. So I put a couple of our resources here in case anyone's interested. We've got our GitHub up there. We've got the CPAC pipeline has a QPP analysis kind of integrated into it now, and then acknowledgements for my lab. So more questions, I hope.
Let's thank our speaker. Thank you. Okay, I'm going to stop the recording now, so we're not recording the official discussion area. And then